we will have a SLO on case control study by Anushree Wagh, 3rd year MBBS. And then our two presenters, Geeti from final year MBBS and Aniruddha Nair by 2nd year MBBS will continue this session forward. By wasting no more time, let us begin this session and embark this intellectual journey together. Thank you.
Good afternoon. My name is Anushree and today I am presenting. Good afternoon. My name is Anushree and today I will be presenting the SLO on the topic case control studies. So these are just some of the topics that we'll be going through. So to give you a brief overview, epidemiological methods are mainly divided into observational studies and experimental studies. And then the observational studies are further divided into descriptive studies and analytical studies, whereas the experimental studies can be randomized control trials, field trials, and community trials. So coming to our topic for today, case control studies. Now case control studies are analytical studies. So they analyze the relationship between health status and indicators. They are used to test whether or not a statistical association exists between a disease and a suspected factor, and they used to measure the strength of that association. Now they are called retrospective studies. So why are they called retrospective studies? Because the exposure and the disease has occurred before the start of the study. So the exposure and the disease occur before the start of the study, and the study proceeds backward, that is from the effect to the exposure. So when we look at the case control study, there are two populations, the cases as well as the controls, and they should be similar in every aspect except the presence of disease in the cases. So for example, say we are looking at smoking causing lung cancer, then the cases would be the people who have lung cancer and the controls would be suitably matched people from the population. And then we would look into their past for history of smoking. Now let us just look at the steps of case control study. First is the selection of cases and controls. When we look at the selection of cases, it's very important to have a basic definition of what cases are with special focus on the eligibility and diagnostic criteria. And we also have to have a definition of where we are getting these cases from, that's the source. And for selection of controls, they should be as similar to the case as possible, except for the absence of the disease under the question. And they should have a chance of developing the disease. What I mean is that if we were studying uterine cancer, then our controls couldn't be met because they don't have a uterus and therefore no chance of developing uterine cancer. Then we come to matching. Now matching is a very important process where we select controls in such a way that they are similar to the case with regard to certain pertinent variables which are known to influence the outcome of the disease, which if not adequately matched for comparability could distort or confound the result. So we come to confounding factors. Now what are confounding factors? They're basically factors which are associated with the exposure but also an independent risk factor for the disease. And so examples could be age, they could be height, they could be occupation, and then we use the process of matching. But something very important about confounding factors is they're very often unequally distributed in the cases and controls. Now the matching process can be individual matching and group matching. Then we come to measurement of exposure. Measurement of exposure we want a definition of what we mean by the exposure. We want to define how we'll be measuring that exposure, and also we want to maintain a uniformity in how we're measuring the exposure in cases and controls. Then we come to analysis next. So here we're looking at exposure rates, and with the example of diarrhea with the exposure being a suspected water source. So what do exposure rates mean? They're basically from the cases, how many people have been exposed to that suspected source. So let's look at the cases. There are 128 cases, and out of those 78 have been exposed. So 78 by 128 would tell us the exposure rate for the cases, and then similarly we can calculate that for our control as well. And from that we can see if there is a statistical association between the exposure status and the disease which So when we look at the diseases or the cases, what are the odds of exposure in cases? That is the number of cases with exposure to the number of cases without exposure. And similarly, we calculate the odds of exposure in controls, and then we calculate the odds ratio. So what is the odds ratio? The odds ratio is a measure of the strength of association between the risk factor and the outcome. To calculate the odds ratio, we have three basic assumptions. That is, the disease should be rare, the cases should be representative of those with disease in the population, and the control should be representative of those without disease. So we get certain important inferences from an odds ratio. That is, odds ratio equal to 1 means the exposure doesn't affect the odds of outcome. Odds ratio more than 1 means exposure associated with higher odds of outcome, and odds ratio less than 1 means exposure associated with lower odds of outcome. So one thing that's significantly seen in case control studies is bias. So bias is any systematic error in the determination of association between exposure and disease. So it basically implies some sort of non-comparability between the cases and the controls. The types of bias are bias due to confounding, memory or recall bias, which is basically explained as a case would better remember their history. So if you're looking at somebody with stomach cancer, they would remember their dietary history better than a control, because they're trying to look for proof of what may have caused their condition. Then we have selection bias, Bergensonian bias, we have interviewer's bias. Interviewer's bias is explained as the interviewer might themselves know who the cases are, and so spend more time and effort in evaluating the case as compared to the controls. Next. These are just some of the advantages and disadvantages of a case control study. Okay, so we're looking at critical appraisal. So why is critical appraisal done? Now, critical appraisal is done to find out the validity and the relevance of the study. So when we're looking at, just to give you a brief overview of this, this is a CASP checklist. This is done for the critical appraisal of a case control study. So some of the questions you can see here is, let's go to question number 3 or 4. Were the cases recruited in an acceptable way, or were the controls selected in an acceptable way? This is basically a question to eliminate selection bias. So they're trying to see if the cases, the controls were special in some way. Were the people who responded different from the people who didn't respond in some way? So that's what we're trying to see. Again, when we look at the 5th question, that is, was exposure accurately measured to minimize bias? Again, they're trying to see if the definition of exposure was given clearly, and was it measured uniformly? Next. So there are 11 questions in this CASP checklist for case control study, and there are basically 3 sections. The first one, that was A, was for what is the validity. Second is what are the results, and the third one is will the results help locally. So can you directly apply it to your population? Can you quantify what the study is giving to your population? And then finally, I just want to discuss a little bit about this study. So this is adenocarcinoma of vagina, association of maternal stem cell therapy with tumor appearance in young women. This was published in 1971 in the New American Journal, and basically any studies before this were based on adenocarcinoma for older women. So this study was conducted because there was a cluster of 8 cases which occurred over a span of 3 years in young women. And so when we look at diethylstilbestrol, that is given early on in, that used to be given early on in pregnancy to prevent premature labor or miscarriage. So they saw, so basically they took the cases, there were 8 cases, and they took 4 matched controls with that. And it showed a very high statistical association between the development of adenocarcinoma in young women and their mothers being given diethylstilbestrol very early on. Thank you.
So you are going to present on this topic,
No, ma'am, this is just a small learning objective. This is the case control.
So what are the advantages of this case control study over the cohort?
Ma'am, it's relatively easy to carry out, and there's a short span, as in a cohort study has to be followed up for a long period of time. And whereas the case control study, basically you're just collecting data, so you get the information quite quickly. And
what about the recall bias?
Yes.
Is it possible with that of case control or the cohort study?
Case control.
Case.
memory bias will be there.
Yes. Any disadvantage of this?
Yeah, disadvantages, ma'am. It's not very good. It doesn't tell us incidence first of all, and it will also not. It's not very good to differentiate between causative factors and associated factors.
You have said that there is a correlation you are saying, na? The correlation is predicted from this case and control study.
Yeah.
So how this is the correlation is predicted?
From the odds ratio, we can figure out the association and the strength of association. And the exposure rates, when the exposure
Maybe
rate,
this is the association, not the correlation.
yeah, it is telling us the association.
So it gives you idea about the prevalence of incidence.
Prevalence.
So what is the difference between incidence and prevalence?
Incidence is the new cases which are coming. Prevalence is the old as well as the new cases.
Thank you, Anushree ma'am, for your SOLO topic. Now I will like to call Geeti Malhotra ma'am for their presentation. Thank you.
Good afternoon everyone, I am Geeti Malhotra and I am a student of final year. Today I will be presenting a paper which is a case control study. So the topic is, can opium use contribute to a higher risk of incidence of colorectal carcinoma? It was conducted in a city in Iran and published in 2016. So I'll give you a brief overview of the structure of this presentation. We'll start with a brief introduction and a background, followed by the methods that they use, which is tools for data analysis, how they collected the data, following which we go on to the results and the discussion, and we'll end with limitations and the critical appraisal of the paper. So we'll begin with the introduction. Starting with colorectal cancers, it essentially includes cancers of the colon, rectum and anus. And to give you a recap, it may be caused by certain mutations, it may be familial, it may result from if you have already a bowel disease, and it may progress onto a carcinoma. Now in Iran, they found that the incidence has been increasing over the past few years, and secondly, usually the mean age at which incidence occurs is 50 to 70 years, but they found that recently incidence has been occurring in people of a younger age group. Moving on, there are also some known risk factors to the incidence of CRCs, which is smoking, consumption of red meat, and obesity. Now coming to the second aspect, opium or opioids. They are a set of drugs which have a high abuse potential, and lately the prevalence of this drug use has also been increasing. They can also be used as analgesics, and they may cause certain systemic effects in your body, such as respiratory depression or coma. Now, opioids are a known risk factor for a few other types of carcinomas, which is larynx, lung, and bladder cancers. And because they noticed that there is a growing trend of opium use and a growing incidence of CRCs, we've conducted a study to see if there's any association between the two. Right, so we'll start with the methods. Coming to the sample size, 175 cases were taken as a sample and 350 controls were taken. So that gives you a case to control ratio of 1:2. During the conduction, so when you're calculating the sample size for a case control study, there are certain parameters you have to enter in, such as an assumed odds ratio and an existing minimum exposure to opioid use in the general population. So those factors were entered. And the cases and controls, as Anushree said, they have to be matched. So they were matched for age and sex. I've just given a quick picture of how the sample size was calculated if you want to refer to it later. Now coming to where the data was collected from. So the data on the incidence cases was collected from a cancer registry of a medical institute over the span of 2 years. For the controls, they followed an interesting method. So they traced these cases down to where the person lives. They set up interview calls, and they picked the first home to the right of the case home and found the first person that matched the age and sex parameters and said, okay, you will be our control. So that way you're also matching the socio-economic status and any other environmental exposure that may have contributed. Now the tools that they used for data collection. It was an interview-based study, so they used a questionnaire. The questionnaire had 3 parts. The first was demographics. So you ask information about age, sex, marital status, education. The second part was dietary history. So as we mentioned previously, diet does play a role in the development of colorectal cancers. So they did ask for diet history, and interestingly they asked for an overall 10-year diet history because that's how long it takes for cancer cells to develop. Third thing is, that's probably the most important part of our questionnaire and what we're actually studying here. Opium use, smoking, and alcohol. The specific factors will come to later. So, data analysis. So after collecting the data to analyze it, what they did was for opium and cigarettes, they calculated a median use. And anybody that lay above that median use was put into a high users group. And anybody that lay below that median users group was put into a low users group. So this was followed for opium and cigarettes. For the third part, alcohol use, it was divided into consumers and non-consumers. Also, for the cases, it was divided into 2 parts, just colon cancers and CRCs, which is cancer of the colon, rectum and anus. Now for the analysis, conditional logistic regression was used. So we calculated the odds ratio and checked to see if there's an association, the way Anushree said. So we'll come to that also later. Secondly, dietary factors were also included and a statistical software was used for analysis. Now we move on to the results. So as mentioned before, there were 175 cases and 350 controls. We'll also explore the results in the similar pattern that the questionnaire followed. So starting with the demographics, we explored the demographic distribution. We saw that, okay, over most of the cases ranged between 50 to 70 years of age. That was an expected result. More than half of the cases were illiterate. And in addition, 81.2% had colon cancer, 17% had rectum cancer, and 1% had anal cancer. So I've just put up the analysis table here. So you have the variables, which is gender, marital status, age, and education. Now, as I said, the cancers were divided into 2 parts, CRCs and colon cancers. So that's what we've done here, each having their matched controls. So the values are provided. We can move on to the next slide. Now we've covered the demographics, we'll cover opioid, cigarettes, and smoking. So it was found that incidence like opium usage was linked to an increased incidence of CRCs and colon cancers. The mean age at which people started consumption of opium was between 20 to 30 years, and the most common method of use was cumulative use. In total, we found that 25% of cases had used opium and 8% of controls had opium usage history. So now, just to give you a breakdown of how the opium use was analyzed or calculated, we look at 3 parameters. The first is your daily dosage or your daily use of opium. The second being the duration of your use, and the third being the current consumption of opium and the cumulative use. A dose-response relationship was established between opium use and the incidence of CRCs. Now, I'm not sure if you can see this at the back, but I have again put the analysis table. So you have the 3 parameters that I talked about, amount of use, duration, and cumulative use. The same way it's been divided as CRCs and colon cancers along with the controls. Now, as Anushree mentioned, to find out if there is an association between these 2 factors, we have to look at the odds ratio. So there is a section on the adjusted odds ratio which I've highlighted. Now, if you look at the values, you'll see that they range from say 3 to 8, all of which indicate an association between opium usage and the incidence of the cancer. Now, after opium, we'll explore cigarette use and alcohol use. So interestingly, it was found that the use of cigarettes did not contribute to the development of CRCs. Now, keep in mind, this is an established fact that it does cause CRCs. It is an established risk factor. another limitation here is there was very low prevalence of alcohol consumption, or rather low reporting of alcohol consumption. So I'll begin with those tables here. You see that the odds ratios are above 1, but they are relatively low. They're ranging from 1.2 to 1.6. Now, Anushree mentioned that an odds ratio above 1 indicates an association. That is true, but it also depends on the confidence interval that you take. So in this study, it's not significant. Now, coming to the discussion part of it, it was proven that the strength of the relationship observed in this study is significant, and opium use increases the risk of CRCs 4.5 times and colon cancer about 5.7 times. The confounding factors were controlled, which is dietary history mainly, and it increased the relationship actually. Also in Iran, opium is usually consumed in 4 types. It's raw opium, burned opium, heroin, and sap. But they only found incidents of raw opium usage and sap usage, so they were both just analyzed together.
You've got 5 more minutes to complete it.
Right. So now, because we're saying that okay, opium usage is causing these cancers, there has to be certain biological plausibility to this as well. So we're going to explore that part of the topic. Yeah. So they did some studies, or previous studies have shown that opium usage has caused mutagenic effects in certain cultured embryo cells or in Salmonella strains. So that is one part of the evidence. Secondly, it may be that maybe the opium is not causing the cancer, but in certain places, this opium that they consume in itself has impurities and additives. And it may be that those additives are causing the cancers. For example, they found that the amount of lead in the blood of addicts was a lot more as opposed to the usual. Okay, this will come to the limitations. Go ahead. Right, so there are certain limitations that the study has put out by themselves. There is a part of critical appraisal, which is my opinion, so we'll go through both of those and then we'll open for questions. So the first limitation is that you can't prove a causality because it's a case control study, you're proving only the association. Second, it's a relatively low sample size. You can't generalize it on a much larger basis. Third, there may be the biases that Anushree talked about, which is a recall bias, an interviewer bias, a memory bias, or a reporting bias also. Alcohol use may have been underreported because it is a social stigma. So we can't fully rely on the results that we did from that. No association was found between cigarette usage and the development of cancers. That is a limitation, of course, because we know it's an established risk factor. It may be because of the low usage of cigarettes among the chosen population. There are certain strengths that the paper also talks about. Firstly, when you're classifying the cancers, right, there may have been errors there, but here it was picked up from a very valid registry. So that is the strength of the paper. Secondly, because of the way that they chose their cases and controls, you know that the confounding factors related to socioeconomic status and environment were matched. Now coming to the critical appraisal. In my opinion, there's an insufficient biological plausibility to this. Yes, they have conducted certain studies on some strains, but I don't believe that's sufficient to actually point to a causation. Secondly, it was done in a very narrow geographical region. It was in a city in Iran. It may be that this can't be applied to other regions because of certain environmental factors that are maybe increasing the relation in that area. For example, the additives in the opium. Third, I believe that a history of a genetic predisposition or family history should have been included because CRCs are known to run in families. There are certain syndromes and certain mutations that cause the incidence of CRCs. Also, I believe data on a person's occupation should have been asked in case of any occupational exposure to radiation or any other mutagenic effects. Fourth, the range of odds ratio is vastly varying. So if you remember the table, the odds ratio ranged from about 2.8 to 8. point that I had in mind was you're asking the patients to self-report their opium usage. So that is an obvious limitation. A suggestion to overcome this would be when you're quantifying the daily usage of opium, something like taking urine samples would have been more precise and accurate and prevent that bias. Of course, it's not feasible for all the parameters, but something like daily usage in a current consumer, that is something that would have been quantified better. Lastly, a strength that I noticed was that they, in the questionnaire that they asked the non-threatening questions first, and then they brought it to more threatening questions. For example, I'll start with your demographics, I'll start with socioeconomic questions, age, marital status, and then towards the end you'll ask about opium usage. That is something that you should consider as an interviewer in a case control study, and that's what they had also followed. So that's the end of my presentation. Thank you. And
First we will take questions from ma'am, then we will move to us audience.
First is that this is a nice presentation, but you have to specify which topic. Meaning to say, if first you have to write the title. You have not written the title of this paper.
the title is the first line itself. Like, can it cause? That's the title.
What?
The first line that was put.
Whenever you are going to present your paper, how you present your paper? So you have to present like this only. Others paper also you have to present like that only.
Yes.
So first slide should be of that the title of this topic, and then you have to write the what are the contributors, mean to say authors.
Right.
So that slide is absent.
Ma'am, the first slide actually I've added it. I just didn't talk about the contributors. Just open the first slide. This is the next one. So this is the title actually. They framed it in such a way. So
Now why you have chosen that this Iran study, not the Indian study?
and the objective was just to choose a case control study so that
So case control study is in India also we are doing.
yes ma'am,
There are so many studies.
it's just I believe it makes for a better learning objective if we have a paper that we can criticize more.
You can take that PubMed. You are
Yes, ma'am.
searching from the PubMed. Huh? On the PubMed also there are some Indian studies which are giving to that better than this. Or this disease is less in India. This is more common in India.
Yes.
And how much should be the odds ratio that should be the proper exposure and the disease? You are saying that the odds ratio is that is equal to the 3 to 8, something like that. So for the proper odds ratio, what should be that range?
So to show an association, the odds ratio that you need will vary from study to study depending on the confidence interval that they choose. Here they had an assumed odds ratio of 2. My point was that because the odds ratio of the different factors under opium usage would vary. For example, it varies with the duration in a different amount as it varies with the daily usage. So what I was trying to convey was that there is a certain discrepancy in the ranges of that. And it's better if, for example, all of them are about 2.3, 2.6 as opposed to 2 to 8.
One more question is that you have arranged or you have to arrange that first is the title. So I am saying that first title, then you have to take that these authors. Second slide should be of that abstract.
Okay.
So what are the points to be included in the abstract? And what is the abstract?
You start with a short introduction of the study. You start with the methods that you use, the sample size, the data, the way of analysis, and a short section of results and conclusions.
So that should be the second slide.
Yes.
And third will be of that introduction. You have given that introduction, but that brief summary you have to give.
Yes, ma'am.
And not each and everything you have to write is of any. You are explaining well. So you have not to write all these things.
Yes, ma'am.
Now, have you written the aims and objectives? What is the difference between aims and objectives? achieve that aim. That is the definition. And in the discussion and conclusion, have you written that the discussion?
Yes.
So in discussion, you have to write that this author has regarding this study, this and this. Say for example, your name is Geetika et al. So whatever authors are there, Geetika et al. And in which year you have studied, and that you have correlated with this. So you have to write that study which is correlated with this. So you have to write the points, point wise, whatever authors have you given. And in the references also, references should be in the Vancouver style. So that was in Vancouver. Do you
Yes.
know about that Vancouver? What is that? Vancouver style.
So as part of the paper, there are certain numberings given after each statement that is research based, and that corresponds to the same number at the end, which gives you the references.
References is that is the correlated with your whatever you have presented that. So but the references the style is there. That is the Vancouver style we are writing. What is that Vancouver style? Vancouver style for the papers is different, and for the books also. Books the reference if you are taking, that is the different. So there is a one Vancouver style. In that you have to write that author's name first. If the authors are more than 6, then you have to write that these 6 authors at all. And after that you have to write the topic, whatever topic is there. Then you have to write the journal name. Journal name के बाद you have to write the year. And after year you have to write the what are the page, volume, issue, and then page numbers. So that is called as a proper Vancouver style. So you have to maintain that also.
Yes.
Otherwise your presentation is very very nice.
Any questions from audience?
Oh yeah, sorry. Any questions?
In the opening, we had a discussion about the high impurities and additives can be present in the opium samples which are there. So, in the criteria that they have included, the exclusion and inclusion criteria, they have never mentioned the sources of the opium from which it was obtained by different people. So, even people in the same locality might be obtaining it from different sources of dealers of opium. So, could that be a limitation to the study of exposure because the lead levels will be increased in addicts? So
Yeah, true. It may be because, as you said, that's a very good point actually, that the quality of opium consumed may be different by different people, and that's not
so
something
even
that
for the
is
same amount, maybe the potency is different. The other one was it was correlated with age and sex, but so do we have to assume that they also correlated it with other factors such as height, BMI? Because those might be, because we saw the obesity was another one which was high correlation, which was previously established. So even with the same age and sex, the obesity levels might vary in the same neighborhood because of the levels of exercise. So is that another?
It was not matched, so obesity and height, weight, BMI was not matched, but I believe it could have.
And another one was the age below 50 was very low in the study. Even though in the opening we saw that it is the prevalence is 5 times and it is below the age of 40.
So it's not, okay, so to clarify, one was talking about incidence and one was talking about prevalence. So as Anushree mentioned, incidence is new cases and prevalence is overall existing cases. So it's established that ages of 50 to 70 have a higher incidence, a higher prevalence of CRCs. But what was concerning here in Iran was that they noticed that the incidence was high in people aged less than 40, which is not how you would expect it to be.
Can you elaborate a little bit on, since we're talking about just case control studies and odds ratios today, can you elaborate how you calculate the significance after estimating the odds ratio? Because you've written a slide up here for how the methodology was done. So can you elaborate a little bit on that?
This one. So essentially when I was going through the study, I wanted to see whether the sample they calculated and the assumed odds ratio makes sense to me. So they've said that there is a certain expected percentage of exposure that you have to begin with, which is, in this case, it was taken as 20%. There is a parameter called study power, which is 0.8 here. You have the study type as case control, and there is something called a confidence interval. And based on the confidence interval, you have something called a p-value, which also will indicate. So, for example, at a confidence interval of 95%, your p-value should come to below 0.05 for your study to be significant. To begin with, you also have to assume an odds ratio, on the basis of which you arrive at a sample size. So when we entered in all of these parameters into like a statistical software, you came to a sample size of 330, which is what they've taken it as. Upon analysis, we saw that the odds ratios range from 2 to 9, whatever it is, and in some cases below 2 as 1.2, 1.6. So those in smoking and such which came to below, we said that that's not significant. As opposed to the higher ones, we said that that association is significant.
Okay, then we will proceed with our next presenter, Aniruddha Nair from second year.
Good afternoon, ma'am. Good afternoon, everybody. My name is Aniruddha from batch 2022, and I will be presenting a paper titled Macronutrients Intake and Stomach Cancer Risk in Iran, a hospital-based case control study. It was published in the Journal of Health Research in Health Sciences in the year 2021. So, an introduction to stomach cancer. So, stomach cancer is the most prevalent type of cancer in the world, and its incidence is about twice in males as compared to females, and increases with age. The latter will play an important role in the study itself. The age standard rates of stomach cancer were estimated as to be 20.6 and 9.7 per 100,000 population in males and females respectively. Most of the cases of stomach cancer are detected or diagnosed while it reaches an advanced stage. So, during that point, therapeutic treatment is usually not useful for the patient, and hence to reduce the incidence of new stomach cancer cases within the country, prevention strategies were decided for stomach cancer control, or to indirectly meaning to reduce the risk factors. So, what are the risk factors of stomach cancer? The major risk factor of stomach cancer is usually Helicobacter pylori infection, or basically peptic ulcers, which usually the main reason why cancers occur is due to dysplasia. So dysplasia means that there is a change in the epithelium of that particular mucosal lining. And Helicobacter pylori causes peptic ulcer, which in short disturbs the continuity of the mucosal lining, which in turn repeated infections can cause stomach cancer. Other factors such as genetic factors, stomach cancer has a genetic predisposition as well. Gender, smoking, drinking, and body fatness is also considered to be risk factors. The World Cancer Research has also added salt preserved foods and obesity as to be risk factors for stomach cancer. But however, it has been noted that there is a limited evidence between macronutrient intake and the incidence of stomach cancer cases. Hence, due to the fact that the results of many studies conducted on the same topic have been controversial. Several studies show positive correlation between dietary factors and stomach cancer, such as the intake of protein, fats, saturated fats, and carbohydrates, where some studies show a negative correlation between protein as well as monosaturated and polyunsaturated fats with stomach cancer occurs. There was a study conducted in Italy which showed a positive association with protein, and which was then again supplemented by a study done in US showing a positive association between cancer and total intake of protein. However, the most recent publication of the cancer research for the American Institute of Cancer research in 2016 indicated that there was no correlation between stomach cancer, or limited correlation between stomach cancer and macronutrient intake. That's right. And hence, given these inconsistencies with different studies with respect to macronutrient intake and stomach cancer, the main objective of the study was to have a defined role between macronutrient intake and stomach cancer occurrence in Iran. So, what is the methodology of the study? The study design was a hospital-based case control study conducted in the Cancer Institute of Iran, located in Tehran. This is also an important factor in the study, since this is one of the biggest cancer institutes in the country within, and around all people from all around the country come to visit this for this particular institute. Hence, we have for their treatment in particular cancers. The study was approved by the Ethical Committee of Tehran University in Tehran University of Medical Sciences, and the study was conducted between 2010 to 2012. The technique used was a structured questionnaire for demographic details. A diet history questionnaire was conducted to understand the dietary intake of the participants. And additional information is BMI, smoking habits which are self-reported at any time last year. BMI was usually considered to be before the onset of symptoms of stomach cancer, or usually before the diagnosis of stomach cancer. And the status software-based analysis was conducted for data obtained by the diet history questionnaire. So what is the participant criteria? The inclusion criteria for this particular study was age more than 40 years. As discussed earlier, stomach cancer has a higher occurrence with increasing age. The next is lack of any pre-existing chronic diseases. The third inclusion is diagnosis of stomach cancer less than 1 year with no history of other cancers. Meaning that there should be a primary cancer within the body itself with no other case of metastasis or any previous history of metastasis. And there should be willingness of to participate in the study. The exclusion criteria were pregnant and breastfeeding individuals, and as an opposite to the third point of the inclusion criteria, with meaning that the stomach cancer should not be there for more than 1 year, and should not be secondary to other cancers. The control criteria consisted of 223 healthy relatives of patients. The patients' relatives were chosen in this particular study because stomach cancer, the main the treatment or any sort of suggestions given by doctors is to do some dietary restriction. So dietary restriction usually affects the whole family, hence they have taken into consideration the relatives to be the controls of the study, and that and they were also matched on the basis of age, number, and residential places. So what is the diet history questionnaire? This is a tool used within the study. It's a type of food frequency questionnaire with 146 questions, each related to the consumption of food and specifically Iranian food in past 1 year. A recall method was used to for the patients and controls to recall that type of food intake before the onset of symptoms, and to avoid under and over reporting Iranian portions of food were used, as well as bread consumption. Why specifically bread consumption? Because bread is one of the most staple diets in the country of Iran, and hence it will become much more easier for the individuals to recollect their diet if compared to bread, or their dietary intake with bread. A dietary supplement consumption was also asked to exclude patients who are on external dietary supplementation, and finally, after collecting all the data, a nutritionist was consulted to re-evaluate and check whether verify the accuracy of the responses. A diet history questionnaires data was then converted into grams per day, which is for the intake, and was then analyzed using the status software analysis. Daily food intakes were then converted into first by using a food composition table. In the study, McCann's and Meadowson's food composition table was used along with Iranian food, supplemented by the Iranian food composition tables, since the latter was not used individually because it only considered special dishes within the Iranian population, and hence McCann's and Meadowson's table was considered along with the Iranian food tables. So what is the McCann's and Meadowson's composition table? It gave a timely and authoritative and comprehensive update of most of the foods consumed by the people of UK. This was considered to be a high standard book for comparing the different food consumption in the world. and they gave a values for a wide range of nutrients such as proximase, dietary, inorganics, vitamins, fibers, and also gave special consideration to food containing vitamin A, vitamin E, and vitamin K. In addition to the analytical data from recent surveys, all these values are continuously being revised on a regular basis, or either on the basis of the food consumed or in the process of being updated. The starter software, the version 14 was used with a significance level of 0.05 and an odds ratio of 1.8. And there was a grouping of participants. According to the study, or the authors of the study, they stated that there might be other confounding factors along with dietary intake, which may lead to stomach cancer, which may influence the dietary intake as well. So they were divided. A multivariate unconditional logistic regression model was employed to calculate to estimate odds ratios and with 95% confidence intervals for stomach cancer. Two models were considered, which were Model A and Model B. Model A was on the basis of age, gender, and energy intake, whereas Model B was on the basis of BMI, BMI, smoking habits, and education. The P4 trend was also estimated using continuous forms of nutrition. So this is the demographic details. An important factor to notice over here is that the education and age and energy intake was significant in the study. In education, you can see the cases were compromised. Higher amount of cases were illiterate, and meaning they didn't have even basic primary school education as compared to the controls. And the age of the cases were higher than that of the controls. The energy intake was also significantly lower than that of the controls. Hence, these three factors also play a role in the type of food which they took or which they consumed on a daily basis. This is a graphical representation of the same data. Then we have different. This was a table which considered the grams per day intake of the food which they consume. In this, these three show a positive correlation with stomach cancer and show a positive odds ratio, positive correlation with the adjustment. This is the cruise odds ratio without considering the models. This was with considering the model A, which was age, sex, and age, gender, and energy, and energy intake, and model B for education and BMI. This is another table which shows with respect to tertiles per intake with the similar results, which showing that sucrose, protein, and unsaturated fatty acids all showing a positive trend with increasing intake. So the general point of all this was that Model A Model B didn't really show much changes with respect upon more adjustment, and Model A showed an increasing showed a highest to lowest with the intake of sucrose, proteins, cholesterol, and calories of proteins showing a direct positive correlation with incidence of stomach cancer. The negative association was also shown between carbohydrates and the calories from carbohydrates. So discussion. So there was a positive and dose response association of the risk with stomach cancer with dietary intake of sucrose, protein, cholesterol, and percentage of calories of protein as discussed earlier, and also calories from carbohydrates also showed a negative association. In this present study, simple carbohydrate intake was associated with a higher intake, association or risk with stomach cancer. And however, there was final conclusion was that there was no significant association was found between total carbohydrates and risk of stomach cancer. Controversial results which were done earlier, through which this paper had taken references from, was a pooled analysis in the USA which showed no association between carbohydrates and stomach cancer. American Italian population study was done in which there was a negative association between carbohydrates and stomach cancer, and a recent meta-analysis was conducted which showed no significant association between dietary carbohydrate intake and risk of stomach cancer, except as shown in the Asian population. The results of the study also demonstrated a higher intake of protein and a higher percent of calories of proteins were associated with a higher risk of stomach cancer. So what was the biological basis of carbohydrates leading increased carbohydrate intake leading to stomach cancer? If it is in short, usually the hyperglycemia induced by the stomach cancer, carbohydrate intake would lead to increased production of reactive oxygen species, which would then lead to DNA damage as well as increased expression of vascular endothelial growth factor, all which favor tumor formation and metastasis. Hyperglycemia also leads to release of insulin. Insulin increases the bioavailability of insulin-like growth factor, which leads to inhibition of IgA binding protein production. And this insulin-like growth factor 1 also stimulates cell proliferation and acts as an anti-apoptotic factor. And the findings of study were, as discussed earlier, were in line with Parry-et-al. Suggestion that stomach cancer was having a positive association with proteins. And however, next slide. And protein restriction was also suggested in patients because there could be decrease in protein intake, which could affect cellular metabolism, autophagy, inflammation reactions, immune responses, and signal pathways, which could also be modified, which also had a second effect as well of modifying gut microbiota, in which recent studies are also taking place for checking the correlation between gut microbiota and occurrence of cancers in the lower gastrointestinal tract. Cholesterol intake showed a positive association with occurrence of stomach cancer, and also this is due to the pro-inflammatory nature of lipoproteins. Next slide. And the conclusion was that it had a potential role in some macronutrients in risk of stomach cancer. And further research with larger samples was also was suggested to be conducted to check whether there was an actual occurrence with cardiac, which is a more common form of stomach cancer, and non-cardiac stomach cancers. The critical appraisal was done using the CAPS checklist, in which the study has addressed the topic in question. The diet history questionnaire and nutritionist verification maintained the accuracy of data collected. The status software also served as an accurate measure to investigate the association of stomach cancer and macronutrient intake, and was updated as per the requirement. The large sample size of 210 was also 210 in provisional participants as cases, and 223 participants as controls. As discussed earlier, controls were relatives of patients and cases, so they were matched on the basis of dietary intake, so there is not much variability in dietary intake. Both groups were treated equally, and division of group into two models on the basis of age, gender, BMI, smoking and habit and energy intake were also increased to the accuracy of the study. There was a comparable socioeconomic position of patients and controls as discussed earlier. Participants were recruited from all around the country, meaning that since their the hospital was a famous one for cancer treatment, and stomach cancer was prevalent in low and middle income countries, the risk factors which were covered as compared to other countries can also be considered. Right? And the study was primarily focused on Iranian diet, which means that this could not be or may be applicable to other diets around the world. The yearly recall method could show potential inaccuracies, since there can be problem in recollecting things from 1 year back or before the onset of symptoms. Also, there is a presence of possible selection bias, since the controls were on a dietary restriction, since they were relatives of the patients themselves. They did not represent the general Iranian population. So, meaning that there can be potential selection bias. And all the food frequency questionnaires may show some sort of inaccuracy in such as misclassifying the type of dietary intake they are on. There can be recall bias, meaning that cases had a higher probability of recollecting what they had before the occurrence of stomach cancer as compared to controls. And there was no official sample size calculation. And the range of confidence interval, as shown in the previous slides, the confidence intervals are very large, meaning that just to make a study more significant, they increase the confidence intervals, and the lower intervals are closer to 1, and with the odds ratio also being closer to 1, meaning that in this particular study, the strength of association is very low. These are the acknowledgements. Thank you.
Nice presentation, and you have covered all these things. That title page is there. There was the table of contents also there. Abstract you have not given in that. In the introduction part, actually you have to explain that what are the macronutrients. Some macronutrients and how they are related to stomach cancer. You have said that, but what are the macronutrients? So you are giving that flow and flow, whatever is given in that. You have to pick up the points, so that you can explain each and everything nicely. And add some diagram on that, what are the risk factor or what are these.
There was a diagram on the right-hand corner regarding the risk factors.
Now one question is that the questionnaire, whatever is there, that was validated or not?
It was a validated questionnaire, diagnostic questionnaire. It's been used in almost all epidemiological studies, conducted within Iran as well as in some studies in the world as well. So it's a validated questionnaire.
And what was that regression model?
Ma'am, they had, since they wanted to compare the confounding factors which could affect dietary intake or maybe directly affect the occurrence of stomach cancer, so they conducted a multivariable regression model in which they compared the occurrence of cancer along with, let's, as I said earlier, model A and model B. So model A, like how they consider age, gender, and the energy intake. So these 3 points can also independently affect, like you can say, increase the incidence of cancer, or they can also affect the dietary intake as well. And to adjust it even more, they introduced a model B, which you could say that the added variables of BMI, smoking habits, and all. So they could, so you can say that this is the accuracy of the study.
Is there any that you are saying that these macronutrients they causes that stomach cancer? You are from this study you are saying like that. So can you advise that there is any social benefit of this study?
Yes,
Social
there can
benefit.
be social benefits, but it's not really being brought into the picture because every single study right now which is being conducted on this particular topic has shown a small correlation between dietary intake and stomach cancer. So, but if there was any social benefit, as discussed earlier, there can be dietary restriction on that particular positive agent. So you can
What
say
advice you will give that to prevent that stomach cancer?
mostly like you can say processed foods, because these people were from mostly from lower socioeconomic status, so and more poorly educated. So they are knowledge
So
on
there should be the balance, proper balance.
balanced
There should
diet.
be the macronutrient as well as.
Yeah, they should be educated on the basis of health as well, and as well as what type of diets they can take.
What is that cardiac and non-cardiac?
There are two
stomach
types of,
cancer? You have not
according to the
explained
study,
before that.
yeah, according to the study, they had told that they had to increase the specificity of the study. They told a larger sample size can be suggested along with to check whether there is any correlation between cardiac, in the part of the where the esophagus enters the stomach, which is a more common type of cancer in this population as well as in the world. And with non-cardiac cancer, which means cancers across the other fundus and the pilous parts of the body, of the stomach. So that's the.
Thank you.
So in this study, so there are few results, but focus on any one of them. How does the study differentiate from when they say that a certain macronutrient is associated with an increased risk in stomach cancer? Did the study take any steps to differentiate whether it is actually that presence of that macronutrient, or is it because of some other micronutrient or something that's commonly present in the foods which are in greater quantities of macronutrients? For example, meats might have more protein, or if there's a specific type of cooking done to a specific type of food. For example, smoking food which can have nitrosamines, it's a known causative factor for some type of gastric cancer. My pathology isn't that strong. So did the study take any measures to account for that? And if not, do you think it's a limitation of the study?
Thank you very much for your question. Yes, the study did not take into consideration any of the external factors regarding the type of food they are eating or any sort of cooking habits which you can say leads to increased stomach cancer incidence. This can be considered as a limitation to the study as well.
Thank you.
One confounding factor which I think they did not adjust for was alcohol consumption.
They did not consider alcohol
They
consumption.
were not smoking.
Yeah.
They haven't considered alcohol. Also, they mentioned that the healthy controls were enrolled, their relatives, were controls, because the dietary intakes of the patients were usually changed due to the disease.
Yeah.
But since they are relatives, it's very likely that, like you said, it's very likely that their dietary patterns will be similar.
Yeah.
So it would have been better if they have taken controls from other.
Yeah, this is considered as a limitation as well, critical appraisal, as I mentioned earlier, because these people do, that is the biggest limitation in my opinion was that because they did not generalize population of Iran, they did not represent that. They only like you say a small segment of their family members which were affected with stomach cancer. So it was like a very small, sophisticated
So the diet actually they are taken before their diagnosis, right?
Yeah.
Like if they were diagnosed 1 year back, whatever food they used to eat 1 year ago, right? Also in your study, during the introduction, you had mentioned that the cancers are diagnosed late. So that means that even before their diagnosis, they were having that cancer, and the dietary history is taken during that period. So do you think, can you comment on how that is affecting the study?
It can affect the study as well. Thank you for your question. This can be also considered as a limitation to the study if you consider like how we do not really have an exact idea of how long the etiology of cancer is occurring in that particular patient. So we can say that that can be that can be a limitation in this particular study, because as you said, dietary intake can be reduced. It can still be the same, or we can say more reduced if taken from 1 year back as compared to, we can say, if the disease has started upon a longer duration. So as you said, diagnosis का आपने बताया